All right, we'll go ahead and get started. Welcome, everybody. Thank you all for being here for the second installment of Introduction to Plant Phylogeny, Understanding Cladograms. In this edition, we'll be talking about examples. Dr. Abbott will be going over specific examples of plant names that have changed, been shifted around, and who's also willing to take questions. He was telling me right before this that he has a lot more slides than he's able to cover in an hour. So if there's anything specific you have a question about, he's happy to accommodate that. We took some requests at the end of the last webinar, and so he will be addressing those as well. We are very excited to have Dr. Abbott back again to share his knowledge and expertise on plant phylogeny. As you all know, for those of you who've been watching the Native Plant Society webinars consistently, he's been a regular in our webinar series, and we're very thankful for all that he's doing. He's not getting paid for any of this. He's volunteering his time to share his love of botany with the masses. So, we definitely want to express our gratitude to him for that. This webinar is being recorded. Afterwards, the recording will be uploaded to our YouTube channel. If you'd like to learn more about the Arkansas Native Plant Society, you can do so by visiting our website. Joining is pretty easy. We try to make it that easy on you. And just go to anps.org/join, where you can use your PayPal account to join online, or you can mail us a check. The address is on that website. The upcoming webinars we have next in our series, Saturday, July 8th from 3 to 4 p.m. at Central Time Zone. Karen Willard is going to give us a program on sedges, the Carex species of Arkansas. And then in August, on Saturday, August 19th, we have Theo Witzel for a 2-hour program starting at 10 a.m. from 10 to noon on natural Northwest Arkansas, the Benton and Washington County Natural Heritage Inventories. So that's a study that they've been conducting over the past few years. And so we're going to get to hear about the results of that. So without further ado, I'm going to turn it over to Dr. Abbott. So Doctor, if you would like to lead the discussion here.
Cool, thanks. All right, so welcome back, y'all. I will stress again that if you've got questions, please put them into the chat or just interrupt in some way. Basically, as I give this presentation, I can't see y'all's faces. I don't know what questions you have. I can't even see very well all the different controls and stuff. So please, if you've got questions, don't hesitate to interrupt. Especially with what we're doing here today, there's 3 times more material than I could possibly hope to cover in an hour. And that's deliberate. I wanted y'all to be able to look back at this material. There's a PDF of this that has been shared, so please just ask. But it's essentially this is we're going to review a little bit from last time, and then answer the questions from last time, and then talk a little bit about kind of some of the pitfalls and why names have changed and such like that. But again, any questions, please let me know. So where we're at last time was we were just kind of going over the basic vocabulary, talking about how cladograms represent hypotheses of relationships, basically. And there's a few basic words that need to be understood. So if you refresh those, reviewed those, hopefully you remember the words as I use them here today. We're gonna expand and talk about a few new kind of different perspectives, reviewing the old stuff, but from slightly different perspectives, and then answer those questions and talk about some examples. That's the plan for today. And so one of the first points I wanted to make here is that when cladograms are being displayed, there's a lot of different ways of laying them out, and it's mostly just artistic. It mostly has nothing to do with the content being different at all. Although sometimes there are certain kinds of cladograms where the branch link, the branch below each of the names, actually has some sort of built-in meaning, whether it relates to, it's calibrated to time or calibrated to branch link differences. But keep in mind that no matter what the layout or format is, the nodes are reversible, and it's really about having sister group relationships. What's most closely related to what? That's what cladograms are telling you. Don't let the display itself fool you, basically. But if you are curious about if the display has any meaning, just check the fine print of whatever article you're looking at, and they should explain if there's built-in meaning. But I will warn you up front, usually there's no built-in meaning. It's just a convenience or artistic reason for why they've laid it out the way they have. And so reviewing from last time again, very quickly again, this kind of modern approach of using cladograms is in theory looking at all the data. I will go ahead and stress that it's actually rare to truly use all of the data in the analysis. Usually the analysis is still only based on DNA with the additional details kind of inferred or mapped on secondarily. There is a stress on these unique shared derived characters, these synapomorphies. But at the same time, I got to remind you that if the sampling has been inadequate, if you're using the wrong data source, you might have what looks like a results that are completely false. Basically, you can be misled if the taxon sampling and the data that are used are incomplete, basically, right? So you have to know the group well enough to understand that again, just because you see it on the page doesn't mean it's absolute truth, right? And then this part about tracking evolutionary history. And keep in mind that again, that is rarely being directly tested or incorporated. It's inferred from our understanding of DNA. You know, the idea that the DNA between close related organisms will be more similar than it is between distantly related organisms, right? There's a certain circularity to this. Just because people put that veneer of evolutionary history on after the fact doesn't actually mean that it was independently assessed all the time. Right? And the final point that we made last time is focus on monophyly. We wanted to kind of stress again that what we interpret as monophyly is a direct artifact of which taxa, which species have been sampled, and what data are being used. So keep that in mind. That even though we've talked about this kind of methodological approach, there are pitfalls along the way, right? And ultimately, when it comes time to apply the results of the analysis, we have to then also accept that at some point rank is arbitrary. Which groups to name and what to name them is definitely arbitrary. There's a subjective element to that, right? So and we talked about that stuff last time. We also then talked about the idea that another way of looking at all this is that essentially what we're doing with these cladograms is we're teasing apart overall similarity. We're no longer letting the artificial convergence fool us. We're no longer letting retained ancestral features fool us. We're going to focus on these shared derived features, right? And so it's essentially wording in a crass or cruder way or whatever is that old saying about if it looks like a duck and quacks like a duck, it must be a duck. Turns out that's not true at all, right? And so our little vampire duck friend here, right? Is it really a duck or is it a vampire kind of thing, right? I mean, the bottom line is sometimes we're misled by data, by appearances, and this approach tries to circumvent that. And so again, visually, I'm just kind of, again, as I said, I'm just reviewing some of the basics here from a slightly different perspective here. Hopefully you're understanding and following. But the idea is, once again, we're going to avoid artificial groups. And so the group in red here is artificial because it's a group that contains an ancestor, but not all of the descendants. So it's artificial, it's non-monophyletic. The group in blue is just convergence, it's false similarity. So if you've named your groups based on the patterns that you see in the red versus blue here, then you're going to have artificial groups, basically, right? And so we try to avoid that. And this is the hardest of why so many names have changed in recent years, is because we're simply trying to make sure that our classification reflects natural lineages, not just human misinterpretation of what we thought looked similar. Right? And so this is where we ended last time. So we're gonna kind of start again from here. Basically, this idea of having a few basic patterns. Most of the name changes issues fall into these four basic patterns. I do want to kind of give us one little warning here, and that is that every generation gripes about all the name changes, about all the change. Modern taxonomy started in the 1750s, basically, right? So 250 years ago, 278 years ago, and names have been changing ever since then, right? We have more than 1.2 million plant names for what, 400,000 species of plants. There's 3 or 4 bad names out there for every name that's accepted, right? So name changes also have to do with the taxonomic rules, the nomenclatural rules. It's not always about there being some biological or methodological error. Sometimes it's just simply that we have rules that were violated, even if the rules didn't exist at the time that the original name was proposed. Somebody else later decides, well, that name is no longer valid. So a lot of the names that are out there that have changed have nothing to do with this monophyly or patterns. It just has to do with nomenclatural rules again, and that's a separate issue. And so at the end of last talk, basically, there was a question about the idea of looking at trees as metrics for the data themselves, the features versus the organisms. And just because you have a phylogeny or cladogram, how do you know whether or not it's got meaningful data in it, right? As opposed to, is it just some sort of artifact of that particular sampling, right? But before I go into answering this question, I do want to stop and just ask, are there any questions? Anybody have any questions for me at this time about the review?
Feel free to unmute your mic if you have a question or put it in the chat.
Okay, cool. So hopefully the review was clear then, and we're all on board. So basically, this idea once again of how do you know whether you can trust a tree or the data in it, and how much is it really reflecting data? I want to talk about this for a few minutes here, because this is a methodological thing sometimes, and there are people that spend years studying this, trying to understand, trying to improve. But bottom line is, if the research behind the tree is valid or good, then the tree does have predictive value, and understanding the phylogeny does have a lot of implicit answers and data in it, right? Should be able to answer other questions if the tree is valid and good, right? But again, keep in mind the garbage in, garbage out, right? If you don't have all the right species sampled, if you don't have the right data, your tree, your results might look good, but be completely meaningless garbage. And the other thing to keep in mind as well is that a lot of times our cladograms are only based on partial DNA data, right? And any interpretation of morphology or evolutionary stuff is actually secondarily inferred or mapped on. And so we're gonna talk about this in more detail, and then we're gonna come back to this. The one specific example that was asked for was Cornaceae, the dogwoods versus the black gums, right? We're gonna talk about them as an example of where the exact delimitation is arguable, basically. Okay? But for starters, though, let's go ahead and kind of remind ourselves of the fact that most modern phylogenetic work is DNA based. And so what we're looking at here is a matrix, a character taxon matrix of some orchid research that a friend did. And the colors represent the four, remember, if you know DNA structure, there's four base pairs, right? Adenine, cytosine, guanine, and thymine, right? So the A, C, G's, and T's. So when you do DNA sequence data, you're essentially figuring out what base pairs are in what order, and then you're comparing the species based on their shared similarities with base pairs, right? So if you look at the example here, their DNA in this particular region here is the same across all the species, except for these last four over here on row 28 or column 28. You can see that these four species at the bottom here have a mutation where the adenine turned into a thymine in those four. And so in this overly simplified example here, what we're saying is that, okay, we're now going to hypothesize that those four species are probably closely related to each other because they share that mutation. Right? There are pitfalls to that, but that's what underlies DNA data as the source for understanding relationships, right? Is the idea that if the DNA is the same, they're probably related in some way, right? But ultimately, can DNA lie to it? Can it mislead us? Yes, of course, right? There's so much we don't know yet. If you think about stuff like what people thought they knew 50 years ago versus 100 years ago, the same thing's going to happen, right? Another 50 years, 100 years, they're going to look back at the future scientists are going to look back at what we think we know and laugh at us for all the ignorant gaps that we have, right? And we already know through the field of epigenetics, the idea that things besides DNA control which genes get expressed. There's a lot of epigenetic things going on here that might actually be confounding factors. We also have to keep in mind that sometimes the genes are linked. So you might have lots of different of data, but it turns out that it's actually all just a bunch of linked data. So it's actually only a sample size of one as opposed to a sample size of 10 or 100, right? Individual organisms or genes can have uneven rates of evolution. What I mean by this is that not all organisms and not all genes evolve or mutate at the same rate. And so when you start comparing things based on how different or similar they are, that might be a false paradigm if you're dealing with organisms that have mutated more quickly than others, right? Remember that word homoplasy, referring to evolutionary noise, false similarities and false differences. Homoplasy exists with DNA data as well. So for instance, we have this idea of what's called orthologs versus paralogs. These are different gene copies. Genes exist in groups called families, all the different mutated forms of the genes. And the ortholog is the copy of the gene that particular individual or organism is using, but other organisms might be using a different copy of the gene. And so if you start comparing the DNA, you might actually be comparing apples and oranges sometimes. You might be comparing different gene copies that are not actually homologous, right? And so those are some of the pitfalls, and hopefully that was clear enough. But if you have any questions, you can follow up on that. Historically, classification was based primarily just on kind of external, internal structure, morphology, and anatomy, right? gross morphology. What does it look like? With the modern paradigm, you can absolutely code these data. You can figure out a way to go through and actually map the data, code them, and run the analysis based on these data. But typically in the modern times, most people don't do that. Most of the time, you're only using the morphology to assess the validity of your DNA results, right? So essentially, you run your analysis based on DNA, and then you say, well, that matches what we understand of morphology, or you'll say, no, it doesn't match what we thought we knew. And when it's tested, there's so much argumentation. Well, if you decided to go ahead and kind of phylogenetically analyze the morphological data, people are going to argue with you about how you assessed homology, basically. You have to assume that structures are homologous. So for instance, if you were running an analysis of things with wings, we know that you shouldn't call bat wings the same as bird wings, right? Well, most of the time, we don't know how to assess homology. If things look the same, we're going to code them as the same feature, but we might be getting lied to, right? So generally speaking, this kind of morphology is it just mapped onto the cladogram after the analysis is over. You don't usually directly use this, and most of the time, for most workers, they're not directly testing morphology. They see it as unreliable. So, for instance, succulent plants, the aloe here at the top with the cactus there, and then we've got this succulent spurge here, and then this carrion flower, this Staphyla awarenea relative down here. So these succulents, they all look kind of the same, but they're completely unrelated, right? And so morphology is seen as this unreliable thing where there's too much convergence, too much false homologies. We just don't understand the underlying patterns, basically, is what people argue. And so trying to assess morphology leads to misinterpretations, basically, is the primary belief. So the question becomes, like, why use DNA? So we've got ourselves a cactus and a spurge here, again, showing these kind of morphologically similar things we just talked about here, right? And so it turns out that sorry, I forgot I had this animated. Basically, this development of the plant body is controlled by DNA. So people are like, okay, instead of relying on the vegetative features that might be lying to you, let's just go ahead and study the control mechanism, the DNA, right? People claim that they're simpler to use and not as complicated. Well, that's true. There's only 4 base pairs, right? So when you're looking at DNA, there's only 4 possible base pairs. It's simpler, right? It's a simpler system. Historically, people use floral characteristics to assess relatedness because that's how plants reproduce, right? And so floral characters were one of the first that were challenged by DNA analysis. It turns out that different plants develop similar flowers because of environmental influences, right? And it's not because of relatedness. And so people said, yeah, we don't want to look at that, right? It turns out that internal anatomy is the one morphological feature that is usually the most reliable phylogenetically. Internal plant structure often tells the same pattern as DNA. We've learned that. My cynical take on this as well, though, is that part of why we like DNA is that we don't understand all the pitfalls. It's still new enough. Keep in mind that, you know, as recently as the 1950s, people still thought that proteins were the hereditary information, right? People didn't know that DNA was the hereditary information until the 1950s, basically, right? And so this is fairly modern for us to start trying to incorporate all this understanding of DNA. And there's so much we don't know. And so I think part of why DNA is so popular is we don't know all the problems yet. So we're learning about them, right? But it gives us a false sense of security, basically. Using DNA just gives us this false sense of security that, we think we understand what we're looking at. And the truth is, it's probably mostly right. I'm not trying to be overly cynical. It's probably okay most of the time. We also use secondary chemistry. And so this is, again, something that's not directly tested. People used to try to test this, but the problem is just because 2 different organisms have the same chemical doesn't mean that they're used the same way. It doesn't mean that they were produced by the same biochemical pathway. As I'm sure you know, there are multiple biochemical pathways going on in every one of your cells at any given second, right? And there are frequently lots of different pathways that can produce the same chemical results, right? And so testing or applying secondary chemistry is one of these things that we use to kind of assess the truth, but in and of itself is also no longer directly tested by most workers. Some people made a huge deal out of geography and saying, oh, well, you can't have a plant that grows in Asia and also in South America. That's impossible. They can't be having babies. They don't have gene flow. And so people used to use geography to assess. but we now also not understand that that's not a directly useful feature. There's no genetic control here. There's no homology to this. It's just kind of random environmental artifacts as to what's where, right? But we still use geography to kind of assess potential issues in an a priori sense, right? So if you find a species that's in Africa and in North America, you might say, huh, that's odd, that's weird. But again, we don't directly test geography other than kind of applying it or mapping it onto the gene trees afterwards. Physiology was something that was used, and again, how do you assess physiology, the biochemical pathways? And this is generally not tested directly. There's too many epigenetic controls, too much environmental influence going on here. But nonetheless, various physiological processes can be used to kind of test the validity of the results, right? So when we say that all data are used, cynical me says, yeah, but not really, right? There's too many arguments about the homology of the data, right? And so a couple examples here, if you think about it, I just mentioned the idea of proteins being hereditary. Up until the 50s, they thought that. Continental drift. I have a copy of a textbook from the 1960s that was still questioning the validity of continental drift, right? Continental drift, people have known about it for 150 years, but at the same time, for the first 100 years, most scientists laughed at the idea that continents move around, right? But we now know that, of course, that's the truth, right? So DNA might seem okay right now, but I do caution us because we don't know what we don't know yet, right? There's a lot to be learned still. I'll stop for a second and ask if there are any questions about that review I just did. Nope. Okay. So wrapping up this kind of introductory review of some of the pitfalls and getting back to the idea of what kind of data is hidden in a tree or useful in a tree is the results of your tree is also then can be influenced by the algorithms used. Keep in mind these analyses are done by computers, right? Some sort of computer language programming has taken place, and what algorithms are used? Well, historically, parsimony, the idea of what is the simplest explanation that accounts for the data, go with that until proven otherwise, right? Also referred to as Occam's razor, the simplest explanation is most likely to be true, right? So parsimony is very intuitive and logical. It's also got its pitfalls, so more mathematically minded people have developed maximum likelihood methodologies and Bayesian methodologies, and these involve predictive modeling and posterior probabilities. There's a lot of math involved. I can't even begin to explain this. I mean, this confused looking little emoji here, that's me, right? But these probability models of evolution are used, but you have to choose the model. Well, how are you supposed to know which model to choose? Well, nowadays the program tells you your data seem to fit this model. I don't like that approach because that's after the fact, right? If you're using statistics, if you're taking your data and you're mining them to find a pattern and then using the pattern to support the analysis, that's very circular, right? So I'm skeptical of these mathematical models. I think that they frequently give you overly high false support sometimes based on the models that they've told you to use, right? But maybe that's just my ignorance about math, right? You know, the bottom line is we do have to be aware that there are a lot of really smart people who are writing these algorithms, and at some point we have to trust them. But a certain amount of skepticism, I think is okay, right? And so ultimately, though, once all that is done, you've got your tree, and you're asking yourself, well, how robust are these data? How robust are these results? Can we trust them? Can we believe them, right? And so we have different methodologies for assessing this. One of them is called a bootstrap support. And essentially, it's this idea of resampling the data by leaving out some of the data and then figuring out, you know, in the different runs, you might run thousands, 10,000 plus, samples where you kind of just randomly pulled out some of the data. And the question becomes is how strongly supported are these results? And so the bootstrap value of these numbers below the line here, so the 100, the 58, so this is telling you that 71% of the time, these two species were sisters. 99% of the time, these three species were related, right? So bootstrap is seen as a way of providing support for the robustness of the hypothesis. But I will remind us that once again, you can get really high support for false results if you haven't chosen the right taxa and the right data, right? Garbage in, garbage out, right? You can get really high support for what we know to be absolute misleading information sometimes, right? We can also then look at the branch lengths as a measure of distance, basically. How different are they from one another? Keep in mind that different taxa might evolve at different rates. So branch length itself doesn't necessarily tell you about relatedness, right? It doesn't tell you what's related to what. It just tells you how different these two things are. but nonetheless, it can be used as a metric. If two things are super, super different, and yet they're supposed to be related, that's a cool story, right? What's behind it? That's something worth looking at, right? And so some of the kind of DNA pitfalls that I mentioned here at the bottom here, this idea of something called long branch attraction. If you've got lineages that are undergoing rapid mutation, right? Lots and lots of changes. You might have different unrelated lineages accidentally converging, showing up as sister to each other, because of all that artificial random change, right? And so we know that long branch attraction has led to some misinterpretation in the past. But again, once we become aware of the problem, we find solutions, right? I will also remind you that there are only 4 base pairs in DNA. DNA just has A's, C's, G's, and T's. The C's and T's, cytosine and thymine, are pyrimidines. They've got a single ring in them, and the adenine and guanine are called purines with these double rings. And so when the mutations happen, basically one of the bases, like a guanine, might become an adenine, or it might become a cytosine or thymine. Essentially, mutations in DNA are base pairs being interchanged, right? The bases being changed. And transitions are much more likely than transversion. And a transition is when a purine is substituted for another purine. So if you get an adenine instead of a guanine, or a cytosine instead of a thymine, those are transitions. And those are way more likely than transversions. So those modelings that I was talking about, the models I was talking about earlier, are based on things like this. They attempt to model basically what kind of mutations are we seeing here, and which ones are more likely, right? And that's what the models are based on, is things like this, right? One thing that's kind of hard to wrap your brain around is that you might actually have a mutation, like an adenine might mutate or change, but still have a new adenine put back in that spot, and it won't be a homologous adenine to the other ones. And we won't know the difference, right? The mutations or changes, because there's only 4 options, might actually end up with something changing but staying exactly the same to our eye, right? Which is again, we're never going to detect that directly without some very specific kinds of testing, right? And then also keep in mind that mutations and changes are not some rare thing. They're very common. They happen all the time. All of our cells have mutations in them. It's just a very, very common part of life for mutations, to happen, right? And so tied to this idea of how the branches may reflect the number of changes, they can also be used to calibrate the time. So the branch length in most analyses doesn't mean anything. It's just a presentation artifact. But read carefully the details. The branch lengths might be telling you how different things are, or the branch lengths might actually be calibrated to time, so that you can actually then approximate the age of the groups, right? So just be aware of that. Sometimes there are data directly hidden in the way the cladogram is laid out. Ultimately, though, remembering this is from last time as well, there's still going to be a subjective element. Which clades do we decide to name? Well, monophily is the criterion that you're not allowed to violate. In the modern paradigm, it has to be monophyletic, right? But the secondary criteria are still argued about. Stability, in terms of like traditional classification, people resist change. So sometimes people argue you shouldn't rename these because that's change, right? Minimizing redundancy. So if you've got something like ginkgo, which is a single extant species, that's a species, it's a genus, it's a family, it's an order. At what point do you want to have overly redundant classification systems, right? Some people let that influence their classification. I like identification tools as being part of my classification, but some people say absolutely not, and that our classification should not worry at all about identification. The predictive value. Some people will then name clades based on which ones are most useful to us in a predictive sense. But are they diagnosable? How big are the groups? There's a push in the modern time, especially with the ferns. If you pay attention to all the name changes in the ferns, that's mostly because they're really ancient lineages, and people have decided, to me arbitrarily, that well, if it's an old group, it has to have a different name than the newer groups, right? They want there to be some sort of evolutionary age equivalency in the names, which I personally don't understand the need for that at all. But that's part of what's driving some name changes, right? And the final point I make on this is essentially that speciation theory. We know that species evolve out of other species. So you wouldn't expect species to be monophyletic, but if species are a rank and you enforce monophily, you are essentially saying species are not biologically real. You're making species an artificial rank as well. But if the higher taxa evolved from species or developed from species and the species started out as non-monophyletic, there are a lot of people that argue that this whole approach is kind of flawed in that way. And then other people do a beautiful job of explaining why, no, it's not actually flawed. It's still a very useful approach. But again, those are discussions for another day. And so wrapping up this first question base, this idea of using trees as metrics for data here. There's a tremendous amount of potential data associated with the cladogram. Most of it has just been mapped on secondarily. From essentially, you have this homology that's implied, which is almost always only based on a limited amount of DNA data. But that said, when you look at a cladogram, if you trust it, if you think the sampling is good, yes, there's a lot of data incorporated in there. But you have to make sure that you assess the data themselves, you assess the support values, and then very importantly, ask yourself what might have been left out. There's always something missing, right? Was something left out because it wasn't necessary, or was it left out just because it was too hard to get? So, for instance, if I'm studying a group that's found in New Caledonia and in Mexico and all over the world, basically, right? And I'm like, wow, you know, I don't have enough money to go to New Caledonia, or I can't afford the permits to go collect this. I'm going to leave this out because it's too hard to get. Well, that's not exactly a very scientific approach, is it? Right? It's logistically an understandable point, but we don't want our science based on gaps that were just because, oh, that was just too hard to do, right? Scientifically, we want to account for everything if at all possible. And so I'll stop here for a second and ask the person that had the question about this, is there any part of this that have I left any part of your question unanswered, basically? Is there any follow-up question? Are you here today? No questions?
I can't recall who asked that question.
It was a doctor from, it was a doctor, but I don't remember his name either. So, but that's okay. If no one has any questions, we can move on. All right. And so again, we don't have time to talk about all these different examples. I will just kind of remind y'all that when I first developed this idea for a presentation, it was part of a course that I taught at a community college in St. Louis, and it was more than 10 hours of lecture, basically. And so the 2-hour version that I'm sharing with y'all is pared down from more than 10 hours of lecture. I left all these examples in simply because I don't know which ones you might find of interest. I'm going to gloss over them. I strongly encourage you, if you see something that you want me to explain here today, just let me know. Or if you just want to follow up with an email later, or if you want to schedule some sort of private chat with you and your friends later on, that's fine too. But ultimately, I'm not going to try to cover all of this. Right? I'm going to gloss over this. We're going to talk about the cornaceae, the dogwood, and black gum example. But I'm not even remotely going to attempt to go through all of this. It's just too much to cover. Oops. I do want to go ahead and remind us of the basics here, that if we're talking about cladograms, once again, it's this idea of talking about patterns of relationship, right? And that what's behind all these name changes is essentially understanding, in a cladistic sense, understanding that all of a sudden we're dealing with groups that are not monophyletic. Any group that is not monophyletic essentially has to have its name changed based on the modern paradigm. So almost all the name changes that the examples are boiled down to a very simple pattern of a cladistic analysis. There's a cladogram that showed that the group we were recognizing was not monophyletic, whether it was a paraphyletic group or polyphyletic. It's a kind of moot point, but they were non-monophyletic, right? If you have any questions about that, please follow up. But basically, when it's time to start applying the classification to the cladogram here, let's remind ourselves that species are arranged into genera are arranged into families, and families are arranged into orders, right? So an order is a group of related families. So when you look at a cladogram like this, I've tagged 3 examples here. Eudicots. Is that a family? Is it an order? No, but it's a higher level group. But is it a class? Is it a phylum? Is it a kingdom or a subkingdom? Is it a superclass? Well, the beauty of this cladistic approach is we don't have to worry about this arbitrary rank. We don't have to argue about what actual taxonomic rank should it be. We can say, you know what, here's a clade, here's a group. The clades are not arbitrary. The rank is arbitrary, but the clades themselves are not. So I don't like getting bogged down in arguments about what rank should it be, right? But when we look at a cladogram like this, understand that if you see that ending ACEAE, that means family, which is a group of related genera. And if you see ALES, that's a group of related families, all right, in theory, right? Well, Amberellacea only has a single species. There's one species in New Caledonia. So if you've got Amberellacea, you actually have just that one species, right? When we look at Aracales, the Aracales here, that's the order that includes the palms. Are there other families in the palm order? Turns out, nope. In most classification systems, there's only the one. I've seen others that propose a couple other families in there historically, but Aracales is just the palm family. So we say, oh, an order is a group of related families. Well, what if it's just one family, right? That's the redundancy we're talking about, right? Why doesn't it just say Ericaceae instead of Aracales? There are no other families in that group, right? So again, you have to already have a certain amount of knowledge about what you're looking at in order to properly understand this. But this idea, once again, that we can then apply our classification based on the cladogram. That's what underlies the naming, right? And so a great example of this, I want to remind us that my reason for talking about this is I'm a field botanist. I'm someone who likes to go outdoors, walk up to plants, put names on them, right? And all this phylogenetic stuff, cladogram stuff, is essentially providing additional support or background, right? It's about understanding relationships, and it can have predictive value for ID. So for instance, this is a group called the Sapindales. It's an order that includes the citrus family, the Rutaceae, the mahogany family, Muliaceae. Semirabaceae is tree of heaven. Sapondaceae includes the maples, and it includes the soapberry. Anacardiaceae, this is the poison ivy cashew family. And Bertiaraceae is the family that gives us gumbo limbo, the gumbo limbo tree. It also is the family that gives us frankincense and myrrh from some biblical stories, right? And so essentially this whole order is characterized by having pinnately compound leaves. So the value of this is if you walk up to a plant that is woody, that has compound alternate leaves, and it's not a legume, and it's not some sort of hickory or walnut, that is, if it's not a Fabaceae or Sapin or Jugulandaceae, it's probably this. You've got like greater than a 90% probability, 95, 99% probability, depending on what part of the world you're in. If you walk up to a plant that is woody, compound, and alternate, that is not a legume or Jugulandaceae, it's probably one of these families here, right? That's why I talk about it. That's why I think about this. This is the value of this is from a practical perspective. You can walk up to plants that you've never seen before and say, aha, I think I know what you are based on your features, right? So this is one of these beautiful orders to learn because it's a very easy pattern, right? And some people are still like, yeah, why should I care about this kind of stuff? Remember, these classifications can be predictive. There can be predictive value in these. So this Anacardiaceae here, this is the family that gives us cashews and mangoes, also gives us poison ivy and sumac, like sumac, where's where pink lemonade came from, right? So this is a family that gave us pink lemonade and mangoes and cashews, but also poison ivy, right? And maybe you should care. So for instance, this is a colleague of mine when I was teaching in Illinois, she shared this this with me. She ate a mango, and this is what happened. This rash that you see on her face and neck here was from eating a mango, right? So if it turns out that you're allergic to poison ivy, you might want to know that mangoes and cashews are related to poison ivy, right? So that you can avoid the potential life-threatening consequences of eating something that causes an allergic response in you, right? Another example of this here, I'm going to talk about this. You can see your Cornelius here at the top here. This is the order that gives us the dogwoods, right? This dogwoods are part of a group called the Asterids, the Sempedalae. This entire group has fused petals. If you walk up to a plant that has a dicot that has fused petals, there's a 99% chance that you're looking at one of these groups here. The only exception that I know of to this in the dicots, I'm sure there's one or two others I'm forgetting about, but the squash family. Some squashes have sympedalous corollas, flat petals that are fused, and squashes are not sympedally, right? But the bottom line is most plants that have fused petals are right here in this group. So boom, this is beautiful, right? So understanding level universality, understanding what that means, right? It can be useful for ID. So within this group here, if you find a plant that has fused petals with these epipetal stamens that are fused, stamens are the male parts, right? Fused to the petals, you're looking at one of these Chorosterids here, right? And within the Chorosterids, you can then start trying to diagnose them. So for instance, the Solanales have flowers that are radial and plicate. The Gentianales have something called collators. The Lamiales have zygomorphic bilateral flowers in a 2+3 pattern. Most families do that, right? So this is the value to me of understanding this cladistic classification approach, is it allows us to all of a sudden learn thousands of species, well, not the species themselves, but to be able to place thousands of species into a group, into a family, by just knowing a few basic characters, right? You can take the traditional approach and go ahead and learn all about the Solanaceae, right? Go in and try to learn about, but I look at this and say, aha, here's Sempedalus, fused petals. It's radial symmetry, multiple planes of symmetry, right? And you see the fold lines in these flowers, these lines in the flowers. When you see that, you've just found either a tomato or a morning glory, right? And so to me, this is the value. And so if you're not interested in the tomatoes or morning glories as a study group, when you see those flowers, you can move on. Or if you are looking for these, that's the way of clenching that you found the right group, right? But that doesn't take away from the need to sometimes go in and learn details about families that go above and beyond a simple cladogram. Gentianales, this is another one of these groups of Sympedalae. This is the family. This includes the coffee family up here, the Rubiaceae, the coffee family, the gentian family. Loganiaceae is the strychnine family. Gelsemiaceae is the Carolina jessamine or yellow jasmine family, right? And the Apocynaceae here are the dogbane family. Down here it says Asclepiadoideae, those are the milkweeds. And so historically, milkweeds were their own family, but we now know that milkweeds evolved out of dogbanes. And so now the milkweeds are a subfamily within the dogbanes, right? One of the kind of warnings that I want to give here in looking at this, and one of the caveats is these family names have been around for a long time, but the genera that are in Loganiaceae have changed. Some of the genera of Gentianaceae have changed. A couple of genera of Rubiaceae changed, right? So sometimes the names are still there, but the genera themselves have moved around. These families basically are not the same as they were historically sometimes, right? So you have to keep that in mind. But this order can be characterized by having opposite, simple, entire leaves with collators. And what are collators? You look at this picture here on the left, you see these little dark triangular structures here on the node between the leaves here. This is a close-up of a branch with young leaves. Those are thick multicellular glandular hairs called collators. And gentianales have them. So do a few other groups. But if you find opposite, simple, entire with collators, you've almost certainly just found a member of this order, which is beautiful, right? And so in this order, we have the coffee family, which is then diagnosable by having interpetiolar stipules. So again, we look here at this branch on the left. We've got the stem, we've got the petiole bases, the reddish structure of the petiole bases, right? And here's this dark purple thing in between them called an interpetiolar stipule. Stipules that are fused together across the internode, interpetiolar stipules. The inferior ovary. So this button bush here. So this is a close-up of the button bush, the upper two pictures here. And then here at the bottom, we've got some of the dioideas, the joe weeds. And so we see here the ovary, the fruit with the sepals on top of it. This is an inferior ovary. If we look at the button bush flowers up here, there's the corolla, and the green stuff down there represents the ovary, right? Inferior ovary. The only gentianales with inferior ovary are Rubiaceae. The only gentianales with interpetiolar stipules are mostly the Rubiaceae, right? There's always a few exceptions, but the bottom line is the understanding the classification and having a classification that reflects phylogeny gives us this great predictive power. I can walk up to plants I've never seen before and diagnose them, right? And the last example here in this Sympedalae here, the mint order. The mints are generally characterized by having these bilateral flowers and specifically having a 2+3 pattern to the corolla, right? It also turns out that the mints and vervains and a few other families tend to have square stems with opposite leaves, right? Not all families do that, but if you walk up to a plant that has square stem and opposite leaves and is sempedalous, fused petals with bilateral symmetry, one plane of symmetry, you've almost certainly found yourself a mint relative, right? Again, the value of this, right? Of course, there are problems, right? But ultimately, once again, I just wanted to drive home that this is what this is about for me. Walking up to plants and putting names on them based on the big picture patterns is what this is about for me. And so we're back to this question of, okay, we've got these 4 basic patterns here. I'll just stop real quick and ask if any questions come up yet.
I have a question. I don't know if it's the appropriate time for this one or not, but I'm wondering about certain, like the Carex genus that has so many different species in there. Is there any reason for having that many species within one genus?
Yeah, so there are only a handful of genera that have 2,000 or 3,000 species globally, and Carex is one of the largest genera in the world, close to 3,000 species basically, right? And so people have tried to split it up. Cobrezi was pulled off, Cymophilus was pulled out. Essentially, people have attempted to pull off different genera, absolutely. But it turns out that Carex, in a broad sense, is just one of these rapidly evolving groups. And as a group, as a clade, it's monophyletic. But when you start pulling out the groups like Cobrezi and Cymophilus, all of a sudden Carex is no longer monophyletic. And so at this point in time, our understanding of the classification of Carex precludes splitting it up. But I'm sure there are people who would love nothing more than figuring out a way of splitting Carex up into a bunch of smaller genera. And so that's kind of where we're stuck with this large genus. And only some of the subgroups are meaningful. Only some of the subgroups are easily characterized, right? But then ask yourself this question: would it matter if Carex are 20 different genera? Then you would just have 20 genera that you couldn't tell apart, right? It would still be difficult, right? It would still be a hard group.
Sure, yeah, they definitely look very similar.
Yeah, and so ultimately, that's kind of what it boils down to: is which clades can be morphologically diagnosed Carex is diagnosed because it has that perigynum, it has that inflated sac fused around the achene, right? There's no other genus of sedge that does that, right? Every single sedge that has a perigynum is a Carex. But yeah, some of those subgroups are absolutely very different, and historically were called different genera. But Carex is non-monophyletic if you pull those genera out. So that's why Carex has remained such a large genus, is because no one's figured out a way to split it up and maintain monophyly.
Thank you. I have one more question, then we have one coming from the chat. Why Polygonum and Persicaria? Why and when did those start getting split up?
So it goes back 200 years, right? The bottom line is even historically, Polygonum was one of those huge genera, several hundred species. That historically they were splitting it up into Tracholon and Antineuron and Persicaria, Polygonum sensu-strictu. So people have known for decades, basically, a century or more, that there are all these distinctive subgroups of Polygonum. And essentially they're being split up now simply because the phylogenetic analyses show that recognizing Polygonum in a broad sense is not monophyletic. It's an unnatural assemblage. It's not a natural group, basically. The Polygonum, in order to have monophyletic genera, has to be split up into the smaller subgroups because they're not each other's closest relatives.
Okay, thank you. Karen Sill in the chat asked, please review those traits of mints again. She said she thinks you said 4 things.
Yeah, so the mint order. So this is where we got to be careful about using word like mint, right? Because you can be talking about one species of mint, a genus of mints, a family of mints. In this case here, we're talking about the mint order. And so the mint order, not all species, but most of our Arkansas mints have square stems with opposite leaves, and their flowers are going to have the petals fused, so they're sempedalous, and their symmetry is going to be 2 + 3, which means there's 2 petals above and 3 below. So if you look head on at a mint flower, it's going to have 1 plane of symmetry, bilateral symmetry, with 2 petals above and 3 below. So if you walk up to a plant that is sempedalous and bilateral with square stem and opposite leaves, you've almost certainly are looking at either a mint or a mint relative like a vervain, perhaps one of the acanths, one of the wild petunias, perhaps one of the other families like a bignone or a scroff or a Polloniaceae. But basically, you're looking at a mint relative when you find square stems, opposite leaves, and sempedalous bilateral flowers. Cool. Any other questions then?
Seeing that we have 12 minutes, I just want to make sure that Shayna has had her question answered. So she had one last time about the dogwood. So if there's anything left to discuss there, just want to make sure we touch on that. Or if you have any further questions, Shayna, please chime in.
Yeah, and so we're on time perfectly because my plan right now was to essentially to end by talking about the dogwoods and black gum.
Oh, perfect.
and then basically just saying, yes, there's a lot more that could be talked about that we don't have time for. Please ask for the PDF. I'm happy to answer more questions about additional groups. I'm happy to schedule a follow-up chat with someone. But the bottom line is, time-wise, we have just enough time to finish talking about the dogwoods and black gum. And so we're going to stop there. But keep in mind, once again, we've got these 4 basic patterns, small lineages derived out of others. Very similar to that, number 2 is the idea of having larger lineages that are evolved out of others, like the milkweeds evolving out of the dogbanes, right? In some cases, the groups were shown to be completely artificial, like the traditional lilies, the saxifrages, the purslanes. Turns out they were completely artificial. Well, depending on how you define them, right? But dogwoods and black gum fall into this other pattern of the exact limitation is arguable. Essentially, there's a certain subjectivity to which groups to name, right? And so whether you're talking about hackberry, is it an elm or not? We're talking about mints and vervains, the nettles versus the mulberries, the carrots versus the devil's walking sticks, the honeysuckles, the mallows, the borages. The dogwoods and black gums are one of these examples of, yeah, there's more than one possible way of approaching this, and it's kind of personal preference, basically. So let's go ahead and talk about this, basically. Cornaceae historically included the Gnisaceae, right? And for some people, they still do, right? The dogwoods, for the most part, have opposite simple leaves. They're always entire. They have these T-shaped hairs. They've got these arcuate venation, whereas Nyssa has these kind of nondescript alternate leaves. And why were they together at all? Well, they both had drupes with ridged pits, right? And they both had inferior ovaries. but otherwise there was nothing morphologically uniting them, right? They were just together based on pollen, based on secondary chemistry, based on, they are closely related, but they were just together because it was a traditional artifact, right? Just to remind you, dogwoods do these kind of cool things. I mentioned those T-shaped hairs here, these little short stalk in the middle. The flowering dogwood has this very distinctive bark. And then there's this thing called the dogwood test. You very carefully break a leaf in half, kind of fold the leaf back and forth and break the mid vein and then carefully pull the leaf apart. A dogwood leaf will actually have the two halves remain attached. There are these spiral thickenings wrapped around the veins. Some Celastraceae do this, a few viburnums kind of do it, but this dogwood test is used by many people to test whether it's a dogwood or not. And if you look at these veins, the secondary veins, the primary vein running down the middle leaf, these secondary veins, the way they're arching smoothly towards the margin and curving towards the apex, that's called arcuate. If you find arcuate veins on opposite simple entire leaves, you're almost certain looking at a dogwood. There are a couple dogwoods that have alternate leaves though, so it gets tricky. Whereas Nissas, the black gums, have alternate leaves. They do not have arcuate veins. They don't pass the dogwood test. And what we have here is this Ogeechee lime, Nyssa ogeechii, and then Nyssa sylvatica up close. And so we look at the analysis, we look at the cladogram itself, and what we find here is that Nyssa, you see here the word Nyssa here, this is the black gums, are distant cousins to the dogwoods. And so there's a side issue here that I'm just going to mention very briefly here. There are people who are now splitting the dogwoods into 3 or 4 different genera. There is support for these 3 or 4 different subclades of dogwoods. Notice that they have 100% support for dogwoods forming a clade. There's no reason to split up Cornus. It doesn't have to be split up. People are doing it because they can, basically, right? There is structure within the genus Cornus, and some people are now pulling it off as several different genera, right? And so these 4 different genera that people are recognizing, the bunchberries, the big bracted dogwoods, the blue and white fruited ones, plus there's other characters. You can morphologically defend these, right? But that wasn't exactly the question that was asked. But I did want to point out that dogwoods might be 1 genus or might be 4, and it's just personal preference. It really is nothing more than personal preference about which clades to name. The question that was asked though about Cornus versus Nyssa here, if we look again here in the upper left where it says Cornus elangium, this is the genus in Asia that looks nothing like a dogwood, and then we have the Nyssoids here. This is the Nyssaceae, right? So historically, the Cornaceae were circumscribed to include all of these different groups here. Well, most people didn't put the Mystixioids or the Curtisias and Grubbias into the dogwood family. So the question becomes, again, rank is arbitrary, which clades to name is kind of arbitrary. Cornus and elangium are sister groups, but they're not morphologically similar at all. The Nissas don't look anything like dogwoods. The Mystixioids don't look like dogwoods. The Curtisias don't look like dogwoods. So at what point do you recognize dogwoods to be like just the 1 genus, 3 or 4 genera, 2 genera, or to include 4 or 5 or 6 different families, right? So this is what it boils down to is that we know what the phylogeny is, but what we don't agree on is the best way of splitting it up. Personally, for myself, I would rather go ahead and recognize the Elangiaceae. So I'm going to go back to where it says Elangium there. I've seen Elangium. Elangiaceae was a family. So, okay, it's a family with only 1 genus. Cornus becomes a family. Cornaceae becomes a family of only 1 genus. But at least they're morphologically diagnosable. What we have now, Cornaceae in a narrow sense, includes Cornus and Elangium, and there's no morphology to define them, right? No morphology to define them. And Nyssaceae currently includes 5 genera that also have nothing in common. And so if you go to the Angiosperm Phylogeny website, Peter Stevens to the Missouri Town Garden, there's not a single shared derived feature to support these families. There are no known morphological synapomorphies to support these families. We have this artifact of sampling, and people are recognizing them as families, kind of, in my opinion, willy-nilly. And there's no reason why you could not continue to have Nyssaceae in the Cornaceae, except that some studies suggest that they're not sister groups. Notice here where we have the asterisks here going on. This is showing that we don't have good support for these. And the squares show, triangle things show even less support, right? So the bottom line is that the DNA do not give us, the phylogenetic analyses do not give us well-supported results. And this is the main reason that people are splitting them out now, is because if we don't really know the truth, it's more useful to have smaller groups than to have one giant group that's a complete mess. But the small groups can't be defined either. So the dissatisfying answer to this question is there's no good reason why dogwoods and black gums are in the same family or not. It's just completely arbitrary. Rank is arbitrary. Right? So that's dissatisfying, but that's also the best we have. So I didn't know if there's any follow-up question about this dogwood thing. Nope. Okay.
Shana says thank you.
Okay, you're welcome. So essentially, what I'm not going to do is now work through all the other sample examples that I had here, right? But basically, just very quickly, just the mints versus vervains. That's one possible thing to talk about. Feel free to look at the presentation and then ask questions. But mints versus vervains was something to talk about. Hackberries versus elms. What's going on there? Right? That was the question. But these are groups when I was working in St. Louis, the nature study group there, the participants in that group, they asked me these questions. These are the groups that your peers in St. Louis wanted to know about, and that's why they're here, right? So hackberries versus elms are now being part of the cannabis family, which kind of boggled everybody's mind that essentially cannabis evolved out of the hackberries, right? Which is kind of really bizarre, right? Mulberries versus nettles was something that confused a lot of people with some of the changes. So if you have questions about that, please follow up. The carrots versus the devil's walking sticks, another versus the Aralias, another question for some. The mallow group, what was happening with the mallows, the Tiliaceae, the Malvaceae, these kind of groups, right? That's one that confused a lot of people. The honeysuckles, what's going on with Adoxaceae, Vibernaceae, Caprifoliaceae, all these name changes taking place, right? Well, that's again, it's another example of kind of personal opinion on how you want to split the groups up, right? And so the other kinds of questions, again, I'm not going to go into the details here. I'm just kind of wrapping up here. These last couple of slides here. Small distinctive lineages, something like corkwood. Well, we have corkwood here in Arkansas, which is why this is worth talking about. And historically, corkwood was seen as its own order. It was the only order endemic to North America. And so even before DNA analysis, secondary chemistry and floral development studies were suggesting that corkwood was just a specialized Semirubaceae, tree of heaven relative, right? And people were pissed. They did not want our only North American order to go away, basically. But the data make it very clear that Lychneria evolved out of the Semirubaceae, right? And so our endemic order went away. Example of these, one of probably the most common issue with name changes are these paraphyletic grades, where essentially groups where distinctive subgroups are being pulled out. It turned out that asters, if you're kind of curious about asters, please read about that, follow up with questions. The spurges, the euphorbias, what's going on there? The hypericums being Clusiaceae, Hypericaceae, what to do? And that's another question. Again, lots of questions here. Don't have time to talk about, but if you're curious about like maples being their own family versus Sapindaceae, the Buckeyes, that's worth thinking about, right? The milkweeds versus the dogbanes, what's going on there, right? This is another example. And then the last pattern that I'm not going to talk about, this idea of completely artificial groups. Traditional lilies, traditional saxifrages, traditional purslanes were all just completely artificial assemblages, right? Traditional workers misunderstood. And it was kind of a shame because the purslanes, for instance, had these beautiful flowers with these 2 sepals on the outside, 5 petals on the inside. It looks like such a natural group, right? But it turns out that the purslanes, essentially the cactus family evolved out of the purslanes, right? And when that was learned, that essentially the cacti evolved out of purslanes, people said, nope. We're not lumping the Cactaceae into the purslanes. And so they split the Portulaceae up in order to maintain the cactus family, right? So kind of personal preference there, right? So if you have any follow-up questions on any of these, please let me know. I'm happy to follow up with you individually or in a group. But I appreciate your time and I hope you got something out of this. Thank you.
Yeah, thank you, Dr. Rabbit. I do have a quick follow-up question real quick. Are there any good resources, whether they be online or textbooks, books in general, references for this sort of information? I'm particularly interested in, which the phylogenetics and some of the morphological characteristics of plants and how that kind of fits in with how they're classified. Like you'd mentioned the interpedial stipule and stuff like that.
Yeah, so probably the two best, if you want an online resource, through the Missouri Botanical Garden, the Angiosperm Phylogeny website maintained by Peter Stevens, is phenomenal. It's also way too technical for most of, even I get confused by the big words being used here, right? But nonetheless, this is an excellent resource online for seeing and understanding the modern phylogenetic information, basically. So I definitely recommend getting familiar with the APG website. And the other part is the other thing in terms of textbook. Let me pull up a picture of this. Judd, there's by Walter Judd et al. Plant Systematics is the single best textbook out there. So this textbook, this Plant Systematics textbook, by Judd et al. is the best go-to reference for in terms of a textbook. So between the APG website and this textbook, yeah, those are the two best references that I can provide.
Hey, thank you. Yeah, that's great. Susan McNutt says, interesting. Thank you. Shana says, thank you so much. This was very fascinating. Karen Seal says, even Dr. Abbott gets confused by the vocabulary. She put a whole bunch of question marks and then a surprised or shocked face. Rob thanks you as well. Yeah, so yeah, we definitely appreciate you. You showed that picture of the purslane, and it looked like one of those leaves were succulents, and so I didn't know if that was perhaps a common morphological characteristics between the cacti and the in that family.
Exactly, and it is absolutely. It turns out that the cacti and the purslane, that's one of the things that tells us that they're related is succulents. Yep.
Yeah, gotcha. So it makes sense as they're evolving. That there's some of those traits that get retained, exactly. Even though other characteristics may change. Yep. So yeah, well, thank you. We really appreciate you sharing your time and your expertise with us, Richard. Want to thank everyone again for being here today. Again, the recording of this webinar will be uploaded to our YouTube channel. If you follow our YouTube channel, you may have already noticed that part 1 was uploaded last Saturday. Hope to see everyone again on Saturday, July 8th from 3 to 4 p.m. We'll learn all about the carex of Arkansas, the CAREX. So that really large genus of sedges that we discussed that has thousands of different species. And that's going to be with Karen Willard, who is a local here in Northwest Arkansas, a local, I consider her a sedge expert. She's very knowledgeable about identifying those in the field. And you can also find out more about the Arkansas Native Plant Society on our website, anps.org. I encourage you to follow us on social media. We're on Facebook, YouTube. I think that might be all for now. And to join, amps.org/join. And I put Dr. Abbott's email in the chat if you want to reach out to him. But just to state it here again, is abbottjr@uamont.edu. That's University of Arkansas at Monticello. So thank you all for being here, and thank you, Dr. Abbott. We really appreciate your webinars. So look forward to hopefully having more with you next year.
Oh, thanks. Thank you all.